Today, further in continuation with the reproduction in bacteria, we will study about the specialized type of transduction. What is specialized type of transduction? In this case, media we use temperate bacteriophage. Upon the infection, instead of causing lysis, the DNA of the temperate phase become incorporated into the DNA of the host bacterium as a prophase. The bacterium harboring a prophase is known as lysogen. The prophase bacteria host relationship is called lysogeny. Prophase may exist in a stable harmony with the host for long period, but upon the exposure to particular environment, stress such as elevated temperature, ultraviolet irradiations, and particularly chemicals, it becomes free. When the phase DNA is excised from the host genome, it excises in an inaccurate a piece of bacterial chromosome adjacent to the prophase is removed and a piece of prophase is left in the bacterial chromosome. The bacterial gene may thus become packaged into the bacteriophage particles which may then infect the suitable recipient. These particles contain both phases and bacterial gene linked in a single DNA molecule, and the bacterial genes are obtained from a particular region of the bacterial chromosome. As these particles contain incomplete viral DNA, they are called defective phase. Upon the subsequent lysis of the bacterial cell, the new phase are released. When such phase infect a new host bacterium, the incorporated bacterial gene undergoes recombination with the host DNA. The recipient accordingly becomes a partial zygote or a merozygote for the gene carried by the phase. Conjugation. Conjugation is the transfer of genetic material from one bacterium to another by direct contact and resembles sexual type of genetic exchange in the higher form more closely. It was discovered in 1946 in E. coli by J. Liderberg and E. L. Tatum in 1958 Nobel Prize co-recipient. It has since been described in a large number of gram-negative bacteria in Salmonella, Pseudomonas, and Serratia. No gram-positive bacteria have been discovered to show conjugation. A clear understanding of conjugation in bacteria came out with the discovery that there is a sexual differentiation in E. coli. Maleness in bacteria is dependent on the presence of extracircular piece of DNA element in the cell called the sex factor, fertility factor, or F plasmid. These cells are referred to as a donor in mating. Female cells lack these factors are called recipient. The F factor contains genes for the production of appendages on the surface of the male bacterium. These appendages are called F pili or the sex pili. These appendages have streaky end that attach to the surface of the female cell, forming conjugation tube between the two cells. The F factor can exist in two different states: the autonomous state in which it replicates independently of the host chromosome and the integrated state in which it is inserted into the host chromosome and replicate along with the host chromosome. A donor cell containing the F factor in the autonomous state is called the F plus cell. The cell which lack the F factor are referred to as F minus cell. A cell carrying an integrated F factor is called an capital HFF for high frequency recombination. Because most experimental work on conjugation has been done with the E. coli bacteria. Cross of F plus and F minus. When F minus come into contact with the F peleus of a male, its strict and conjugation tube is formed between the two cells. During conjugation, the F factor replicates inside the F plus donor cell and one copy of it remain in the F plus while another in almost always transfer through the conjugation tube into the F minus cell, which also become F male. In this process of conjugation, the DNA of the chromosome of the male is not transferred, therefore the genetic recombination does not take place. For this reason, those male in which the F factor is present in a cytoplasmic state are called infertile male. The only genetic alternation effect by the introduction of F factor into an F minus cell is a conversion of F minus cell to an F plus cell. The high frequency high, high frequency F plus cell in certain male F plus cell, the F factor leaves the cytoplasm and integrate itself into the host chromosome. A cell type with an integral F is called HFR. The cell transfer their chromosome gene into the recipient at a very high frequency, hence the name HFR. When conjugation occurs between the high frequency cell and an F minus cell, the F plus cell chromosome with an integral F factor replicates so that one parent strand separate from the other. This parental strand of chromosome is nicked at a specific site or the integrated F factor. A small piece of the F factor leads this strand to the F minus. Thus, the portion of F factor is to be transferred. Chromosomal genes are transferred next, and the remaining F factor, however, in the last segment to be transferred. A portion of the high frequency recombinant chromosome is transferred between the cells. Separated almost always occur between the final segment of the F factor is transferred. Meanwhile, a new strand is formed repairing the high frequency recombinant chromosome. Thus, the male still contains a complete set of DNA sequence even after donating a fair amount of DNA to the F minus. The DNA strand that has entered the F minus now replicate, becoming a double-stranded. It is immediately followed by the recombination of the donor and the recipient DNA inside the recipient cell. Thank you.